Okay. So, good morning to everyone, and a very warm welcome to you all on behalf of SilicoScientia. I'm delighted to welcome you to today's webinar on AI in drug discovery. Myself, Mahima Kolpe, will be your host for this engaging session. Today, we will be exploring how artificial intelligence is revolutionizing the field of drug discovery and how next generation tools are making research faster and smarter. So, in our very first session, that is about artificial intelligence, redefining the future of drug discovery. So, it is my pleasure to introduce our distinguished speaker for today's webinar, Dr. MD Ataul Islam, founder and head of SilicoScientia. Dr. Islam is globally recognized expert in computational drug discovery. Dr. Islam holds a PhD in pharmaceutical and fine chemical technology from the University of Calcutta and brings over 15 years of global research experience, including prestigious fellowships such as Newton International Fellowship, UK, and the NRF Postdoctoral Fellowship, South Africa. Before founding SilicoScientia, he worked as a senior computational chemist at 3 Bigs Omiscore, Bangalore. Dr. Islam has served in various leadership roles at international institutions like the University of Manchester and the University of Pretoria. Dr. Islam is a prolific contributor to the field of computational drug discovery. His research covers a wide range of topics from QACR modeling, molecular docking, and pharmacophore mapping to advanced AI driven screening, molecular dynamic simulations, and inhibitor design for emerging and neglected diseases. Dr. Islam has over 50 high impact peer reviewed publications spanning various journals. Today, his talk will walk us through the transformation of drug discovery in the era of artificial intelligence. Over to you, sir.
Okay. Thank you, Mahima. Good morning, all of you. So today we'll talk about how the AI and machine learning impact drug discovery and how people are thinking the new dimension to discover, identify, or design the new molecules for therapeutic applications. So let me share my screen. My screen is visible?
Yes.
Okay. So we all of us know that is the drug discovery is very complicated process. So it starts from the identification of disease, followed by the identification of mechanism of action, how the disease spreading, and once it is understood that is what is the mechanism of action, that then we go for the target identification and validation. And once you have the target identification validation part completed, you have to find out some new potential molecules. And those molecules, uh, just a minute, I have to take a call. Hello? Yeah? Yeah. Can you hear me? Ma'am, I just send him the details. Okay. Or you may call him or okay. Sorry. So the disease identification followed by the identification of the mechanism of action, or understanding the mechanism of action of the disease, then identification of the target, and followed by the molecule design and identification, and then finally for the preclinical testing, in vitro, in vivo, and then clinical trials, and all these things and approval. So I'll talk in the next slides. Actually, so this overall the competition the drug discovery is a complex process, and we'll see how it is the success rate also. So if you see the traditional drug discovery in the development phase, so you have to understand the clinical need assessment, and the, as I mentioned, the target identification and validation. And once you have the target identified, then you have to mm, uh, find the hit molecules. Here actually you need the screening, and further once you have the potential molecule, then you go for optimization, and then you go for some animal studies. So this is the overall development phase, and then after that you have to go for the regulatory, like preclinical studies, there are good laboratory practice or good manufacturing practice here, clinical trials, and then followed by the different phases. And here the investigation, new drug, or the FDA approval and new drug arrival. So all together, this entire process is very, very complicated, and the failure rate is extremely high. So if you see here, so that actually pre-discovery here, and here actually this is stage is the drug discovery phase, and that once you have to understand in the pre-discovery, like uh, that understanding the disease, understand the mechanism of action, and then target identification. Once you have, then you have to go for the discovery phase to identify the potential molecule. And once you got it, and then go for the different stages of the clinical trials, and like that. So this overall process, it takes around 12 to 15 years of time, and approximate time, and 2 or 3 billion US dollars for a single drug. So you can understand that is how the traditional drug discovery is really complicated and time consuming. So that I mentioned that the cost is high, around 2 to 3 billion US dollars for investment of single molecule, and definitely the time is a very important matter, because it takes, if you have the good funding, you have the infrastructure, all that it may take around 10 to 15 years of time, and success rate is really very low. So some people have reported that if you started from 10,000 molecules, maybe one molecule will come at the end as a potential molecule. And also experimental validation. So these are the main or major challenges in the drug discovery. In this situation, what actually we need the different computational approach in the drug discovery field. Okay, so these are the existing computational drug discovery approaches. Like in case of the target identification, you can go for the database mining. A lot of databases are there, so there you can go, or you can fish the target, like reverse docking kind of thing, or maybe you can go for the different computational method like homology modeling to design your target protein. So that actually the help in the target identification. Followed by like lead identification, there's a lot of approaches are there, like ligand-based approach, or the structure-based approach, or maybe de novo design. So molecular docking, the de novo design, and the molecular dynamics, these are well-known approaches in the computational drug discovery. And definitely the artificial intelligence helping a lot. I'll talk today a little bit about it, how we can apply artificial intelligence and machine learning in the computational drug discovery to accelerate the drug discovery process. Also the multidimensional QSAR, pharmacophore modeling, and in case of the lead identification, maybe the virtual scanning, the pharmacokinetics, biostatistics, or maybe the different molecular properties analysis or triglyceride assessment, and in a preclinical test, the computational is majorly involved data analysis. So if I say that is that your drug discovery, initial drug discovery is very slow, it is time consuming, but if you apply the computational approach, maybe it will accelerate, like you can accelerate this turtle through the different computational approach. So now what I explained to that to till now, that is that traditional drug discovery is no doubt it is a very complicated process, and that when the computation is smart technique or intelligence, that like computational systems came in the power, the drug discovery went up. It got the speed, but still there is a lot of limitations are there. So that I'll talk, that is how these limitations when we can overcome by application of the machine learning or the artificial intelligence in the computational drug discovery approach. So what is machine learning or artificial intelligence? The artificial intelligence is a branch of computer science that study machine learning, machine intelligence. And these are the well-known applications, like if you're searching something in your Google or any search engine, how it is fast. You are giving some keywords based on that, the huge information. And also the Netflix or YouTube, like if you're searching some videos, so it is automatically suggesting the similar kind of videos. That is, they are trying to understand your taste or your choice, and then they will suggest the new videos, or they are suggesting something which may you like. Similarly, the self-driving vehicles, now the hot topic here, automated language translation, facial recognition, computer games. So these are actually the major fields the artificial intelligence are majorly applied. And here also we are the computational people, they're also applying the artificial intelligence in a large scale to enhance the drug discovery. So if I just define another way, the artificial intelligence enhance the human decision making in drug discovery, but does not replace the expertise of scientists. Definitely, actually, if you know, that is with the experimental approaches, you have a lot of limitation. You can explore or you can go for certain limited chemical space exploration, and that actually enhance in the computational approach. That if you have the computational drug discovery approach, your chemical space will be much bigger, but still the limitation is there. So here artificial intelligence actually give you, it will based on the maybe feature extraction or maybe other approaches, it will help you to take decision, but it does not mean that it will actually replace the human being. And another approach that is called this most effective approach combines the human creativity and domain knowledge with the AI speed and pattern recognition and predictive power. And this definitely AI reduces the cost, shorten timelines, and opens possibilities for the personalized and the precision medicine. So these all the complicated things that actually nowadays become very easy when the artificial intelligence and the machine learning involved. So again, the artificial intelligence is the simulation of a human intelligence in machines to perform tasks such as reasoning, problem solving, etc. And the machine learning is a part of artificial intelligence, and that enables the system to learn from data, improve the performance without explicit programming. So this machine learning will understand your data, the known data, and it will train the model. And once the model is developed, that model you can use for further application. So we'll talk about more about this. And further, the deep learning, it is the part of machine learning, and which is a multi-layers neural network to analyze the complex data pattern. So if I'm not wrong, so it mimic the human brains kind of thing. So these are actually the major applications in the drug discovery. So here you can see that artificial intelligence, the machine learning is part of artificial intelligence, and deep learning is the part of machine learning. So depending upon the available data, depending upon the your project objective, you can decide which will go. So I'll talk about that also. So this is actually there are different domains of machine learning, like unsupervised, supervised, reinforcement learning, and they have their different subcategories of each the domains. And these the big question is that when and what situation, what which domain I should take and which branch of this machine learning we should take. So that totally depends upon the availability of your data, kind of data, and what is your research objective. So sometimes we need to go for the dimension reduction, sometimes we need the clustering, sometimes we need the classification, sometimes we go for the regression analysis, or maybe sometimes we go for the real-time decision, or like skill acquisition or learning task. So totally, the machine learning or the artificial intelligence, it is a very, very huge field. Rather, I can say the this is a ocean. So you have to select the domains, appropriate domains based on your data available and what exactly you would like to do. So in a different, along with all the fields, nowadays AI is applying in each and every field. So whenever from the morning to evening and evening to morning in our daily life, the AI is all around. So suppose in case of the game or the mobile application, everywhere we are actually surrounded by our artificial intelligence. As I mentioned, if you're watching the YouTube or Insta, the one or two videos if you watched, the similar kind of video will come for suggestion. So that actually everywhere are applicable, applied artificial intelligence. Similarly, in the artificial intelligence also applied in the different fields of healthcare, like clinical trial design, the quality assurance, quality control. These are the things actually we can do here. Pharmaceutical manufacturing, pharmaceutical product development, pharmaceutical product management, and definitely it is in the drug discovery field. So today's our topic, that is how can we use and what are the applications of machine learning in the drug discovery field. So the artificial intelligence can be used in the drug repurposing or target identification validation, designing the molecule or screening the molecule from the databases, and optimization history also, or biomarker discovery, clinical trials, or maybe these are the things here who can where artificial intelligence can be used. And also definitely it reduces the time and cost, and most importantly you can explore the huge uh, you can explore the huge number of the chemical space. As much as chemical space you can involve, the more information will come, and that will help you to take the proper decision from your data. So I talked about the machine learning thing, or artificial intelligence. That means the data, because from the known data your machine will learn, and that learning and that knowledge can be used for the unknown data. So first of all, you have to give the proper data. Based on the data, your model will develop and the quality of the model will develop. So for drug discovery, from where you will get the data? For drug discovery, the main data is a small molecule, if I'm not small molecule or macromolecules. So from the different databases, you can extract your molecule like Campbell, Pumpkin, Drug Bank, Zinc, Coconut, Enamine. So a lot of the databases are there, or you may have your own database. Maybe you have designed some molecule, you have crystallized some molecule, so those molecules you can use. So along with this molecule, we need some the endpoint that is called the biological activity. And once you have the data, and that data will give some information, that is the properties, that what kind of molecule it is. Suppose I'm giving an example, like suppose you have 2000 molecules, and those molecule binding to the certain receptor like kinase. So I want to know what are the characteristics or features or properties make them the kinase inhibitors. So that extraction actually in terms of we call the descriptors, we can extract the features or the descriptors or the properties in the form of the zero dimensional to the multidimensional form. So 0D, 1D, 2D, 3D, 5D, and so on. And once we have this data, that is the features or the descriptors or the properties, those we can use in the machine learning algorithm, like maybe the based on the your data regression or classification or the class dimension reduction. So here it is very important what, which approach you have to select. So that will tell by your data available as well as what exactly you would like to conclude or what exactly you would like to find. So once you have, then you can use in the different drug discovery approach or the physicochemical analysis or the clinical analysis or the pharmacokinetics analysis. So this is all our, that is the single slide can explain the entire thing. That is the how you can implement the artificial intelligence or machine learning in the drug discovery field. So you need some known data or the structure of the molecules. Either if you would like to go through the small molecule, you need the small molecule. If maybe you would like to go for the macromolecules, that also you can collect, because your ultimately your molecule will go and bind at the active site of the protein. So this way you can actually get the data and develop your model and further go for the designing or the prediction. So these are the some well-known application, but these are not only. This list is more. So here I'd like to explain it here. The de novo design, that is the designing of the novel molecule. So you have to some molecule, from this molecule you extract the features, you design the develop the model, artificial intelligence model or machine learning model, and then from that model can guide you to design the new molecules or new small molecule candidate. Similarly here, like drug target interactions. So suppose you have your protein ligand complexes and binding interactions data, you can extract those data, develop the model, and then you have the model, and that model can be used to the new data set of the protein ligand without further going for the any help of external tools. So that way also you can do for the drug target interactions analysis. Drug target binding affinity, like molecular docking, that is ultimately the molecular docking where what we do, we just understand how my molecule showing the affinity to the towards the receptor. So from docking, we can get the data, the binding energy and the molecular features, and then we develop the model, and then we can predict those molecules in the unknown new molecule. So this is very, very helpful. So I will explain in a short, that is how the docking, how the machine learning is helping in the docking to uh, predict the binding affinity of a large number of molecules. And here, this is the very, very helpful approach in the target structure prediction. So until unless you have the target structure, so you can't go for the structure-based drug discovery field in the computational. So we need the 3D structure of your target. The major source of experimental target structure, 3D structure, is a protein data bank, but some structure still are unknown. So then you can use the machine learning to convert your protein sequence into the 3D structure. So I think all of you heard about AlphaFold. That is a tremendous work, a tremendous job that they have done. All the sequences available till now, they have converted into 3D structure using the artificial intelligence. So this is the first time in the ever in history that the computational chemists got Nobel Prize due to this thing, that is they have converted the sequences in the 3D structure of the target using the machine learning. So these are the few examples. I'll show you some more approaches here. So here, in case of the pharmaceutical analysis, suppose we would like to go for the toxicity prediction. So you should have the known molecule toxic and non-toxic. So understand why these molecules are toxic in nature, why these are molecules are non-toxic in nature. Those features, those information will try to understand, and then based on those features, we can go for model development, and that model can be used for the further prediction of the new molecule, whether it is toxic or non-toxic in nature. Similarly, bioactivity. So from known data set, we can develop our machine learning model, and that model can be used to the unknown molecule, whether it is anti-cancer, or antibacterial, or antiviral molecule. So that will also help to identify the type of molecule before going to the experiment. And physicochemical properties like drug likeness or all these things, or the solubility, or the brain barrier, or drug likeness, or hydrophobicity. So these things also we can develop from the known data set. Once the model is there, you can use the large number of data set in a very small time to predict all these physicochemical properties. So these are the way we can use. So overall, in case of the drug discovery, or in case of the physicochemical pharmaceutical analysis, we need some known data. From the data, we can extract the information, and that information we can develop the model, and that model can be used for prediction for the unknown molecules. So these are the main approaches, or the machine learning technologies are used, like machine learning, deep learning, natural language processing, the NLP, reinforcement learning, generative AI. So depending upon the research objective, what exactly you would like to do. So suppose you would like to study the pattern of the large data set, so you can go for machine learning. Suppose you go for in a deeper insight, and so that you can go for this is yes. Hi, ma'am. Am I not audible?
Sir, now you're audible.
Okay. Okay. My presentation is visible.
Yes, sir.
So which presentation the last which slide?
Yes, this one.
This one is I explained this one or not?
Yes, sir. Yes.
Okay. So that I mentioned, that is depending upon the data, depending upon the research objective, we can select the machine learning method. So here, how the traditional methods, when you apply the artificial intelligence, may be enhanced like that. The target identification, it is slow and manual experiment required, but here you can go for a very fast. Similarly, screening, optimization, preclinical, clinical trials. So all over, you can, if you use artificial intelligence approaches, so definitely it will reduce the time very significantly, as well as it reduce the cost in a large extent. So as I mentioned, you can use target identification and validation. Like you can use the omics data, or you can go for the prediction of disease-causing genes and proteins. So in both data, you can apply your machine learning to find out the target for your disease of interest. So as I said, that is how docking is impacting when you have the machine learning in your docking method. So suppose I'm giving an example. Suppose you have 10 lakhs or 12 lakhs molecule to dock for a certain target. I'm not saying that it is not possible, but it is a little bit difficult. You need a very good infrastructure and you need a time. Suppose 12 lakhs molecules, or maybe suppose 60 lakhs molecules you have. So 60 lakhs molecule, if you're going to dock, so it will take the long time, as well as you need the high computing system. So here, how can we use the machine learning or the artificial intelligence to faster the docking approach? So suppose 60 lakhs molecules you have, you just select 60,000 molecules or maybe 50,000 molecules. So compared to the 60 lakhs molecules, the 50,000 molecules docking is easier. So I'm docking the 50,000 molecules in my normal system, and after 50,000 docking, I will have the binding energy as well as the molecules. So we'll extract the features from the molecule and we'll use the binding energy to dock the data, and then we'll develop the model. Once you have the model, and that model you can use to predict the binding energy of the remaining molecules, because I am using the same target for which I am going to predict the binding affinity. So from 50,000 molecules, I develop the model, and that model I can use to predict the binding affinity of the remaining molecule. So no need to dock the remaining molecule. So it is not only 50 times, I would rather say the 100 times faster compared to the existing. So this way we can use artificial intelligence in the molecular docking. Similarly, in screening, you can use millions of molecules you can screen through the machine learning model, or you can use a generative model. You can design the new molecules with your desired properties. And in our company, we have already implemented these approaches and already published the paper. So how can you use the generative AI to predict the molecules, or how can we develop the model to screen the large number of molecules. Similarly, you can use the AI in the pharmacokinetics analysis, the prediction of the pharmacoproperties, reduce the toxicity, minimize the animal testing. So these are the things actually we can do in our machine learning approaches. Am I audible or not?
Yes, sir.
Audible, sir.
Drug repurposing, that is, what is the drug repurposing? I think we all know that is, if you already existing drug you can use for the new disease, so that is called drug repurposing approach. So here you can use the biomedical literature, analyze the clinical data, explore the molecular pathways. By this, you can develop your model to screen or identify the existing drug for the new disease model. The de novo design, that is designing a molecule, the novel kind of molecule using the machine learning, that is called generative AI, and that I mentioned already that we have a few articles already published. The last year from our company, we have used huge extent this kind of approach in our organization. Also, you can use machine learning or artificial intelligence in the clinical trials, like regulatory submission, data analysis, data management, supply management, trial management, study setup or study design, etc. You can use here. Also, in a artificial intelligence and the big data, how actually helping the drug discovery, like electronic record of the record, metabolomics, genomics, proteomics, medical imaging, AI integration. So these are the broader picture, a broader aspects, a broader field in the AI and machine learning, in the directly or indirectly helping the drug discovery. So overall, I'm telling you that is a machine learning in drug discovery or machine learning in any field is the game of data. If you have the data, you are the champion for to develop the model for prediction. So please are medicine. You can go for the patient-specific prediction, cancer therapy, or rare disease. In all these fields, you can apply the artificial intelligence to design or identify some medication for the patient. So these are some publications. I think around 20 publications we have done using the machine learning different approaches, like degenerative, that is the skinning of the molecules with the machine learning, or docking of the using the machine learning, or the designing the new molecule with machine learning. So these already we have done in our organization. So what are the advantage of the machine learning over the traditional computational drug discovery? Definitely faster and cheaper. Yeah, definitely the one-time investment, and definitely, as I mentioned, in case of the docking event, it is very easy to predict the binding affinity. Once you have the model from the doc data, it won't take much time. Maybe a few millions of compounds you can predict in a less than 1 year or 1 hour. So accuracy is high. Why accuracy is high? Because, as I mentioned, due to the involvement of the machine learning, you can consider the large chemical space. So as much as chemical space you can involve, the accuracy will increase. Definitely the big data handle, novel molecule generation that I mentioned here, target identification that we also discussed, the repurposing, then the optimization, etc., and the risk reduction. So these are the overall few of the advantage of the machine learning. How can I use this here? Now I'm going to the different direction, how the different the field of the drug discovery impacted, and how the company are involved or the investors are involved in this field, and that totally impacting the job market in this field. So the concept of the AI developed in 1950s, and then with the help of with the increase of the data size and the processors, the different computational drug discovery came in the market, like QSAR, computational drug discovery, docking, variable selection, then artificial neural network, the validation application domain, deep learning. Now more and more. So this is actually 2010 data. Now it is far beyond. So now you can see, if you imagine, the people are carrying the mini laptop or mini computer, high configured computer in your pocket in form of the mobile. So you have the high processor, you have the high memory, you have the high hard disk. So with the development of the computational technology, the different computational drug discovery came in the market, and now due to the multi-GPU application, now machine learning is enormously using in the field of drug discovery. So if you see, this is the 2023, this is actually created here, this data. So in a different disease model like oncology, neurodegenerative disease, or metabolic disease, or the cardiovascular or infectious disease, how the people are investing for the artificial intelligence-based drug discovery in this disease model. So exponentially increasing. So by 2030, how you can imagine the people are investing, or the market size is increasing of artificial intelligence drug discovery. So people are crazy to invest in this field because they got the beauty, they got the essence of artificial intelligence and their results in the drug discovery field. Here, in the drug optimization or repurposing or preclinical testing, as other people are the global market size. So in 2023, the total market size was 1.5 billion US dollars. Now it is going increase and increasing. So if you see this circle, so if you zoom it 400 percent or 500%, even I'm not sure that whether you can read the name of the companies or not. So these are the companies or organizations, they are currently involved in the AI-based drug discovery field. So it is roughly estimated that around 4,000 companies, 5,000 investors, or they are involving, they're investing, they're working in the field of drug discovery. So it is the data of 2023. Now, with the time being, that it is growing exponentially. So once the investors investing their money in this field, once the companies are working in this field, so definitely the job is creating the job is much more in this field nowadays, or maybe in the coming few years. So these are the real success stories for how the artificial intelligence involved in drug discovery and the molecules and the different clinical phase. So like 2022, 2022, the two molecules here, 21, 22 here. So these are the molecules is already under the clinical trials in the different phases, and these molecules develop the help of artificial intelligence. So here, the details about those molecules like BXCL701 by the BioXcel Therapeutics, and it is using the drug repurposing approach with the help of the AI. Similarly, Ben2293, the relevant AI, it is a very good company in the artificial intelligence drug discovery, and they have used the machine learning on the multiomics and literature mining, and it is in the clinical trial second. Reg 44881, this is a BioHive company developed, and they have used the structural modeling using the AI machine learning approach, and this is in a clinical trial third phase. BXL 501 BioXcel, and this is they use the artificial intelligence implemented repurposing of the CNS active molecule, and this is also third phase. Baricitinib, this is the Bioinformatics again the company. They use the knowledge graph and AI pathway analysis to develop this molecule, and it is in clinical trial phase 4. And another molecule by the Abcelera Biologics, and this is their is an antibody and developed by the machine learning, and it is also in a stage 4. So these are the known molecules. That is this is the open the data. These are the information opened. That is, these molecules are developed by AI machine learning, but lot of molecules are already under process, maybe in the preclinical testing or the clinical trials, the different phases using the machine learning. So I'm just trying to emphasize here that is, yes, it is not only the story, it is the reality that how the machine learning or the artificial intelligence are actively involving in the development of the real-time molecules, are they are already in the clinical trials. But there is some challenges are there also in the AI drug discovery, like as I mentioned, the data is the main thing for your machine learning approaches. So if your data is wrong, your model, the quality of your model will be compromised. And lack of interpretability of your model also hard to take the decision. Then ethics and regulatory hurdles are there. Need for multidisciplinary collaboration is required. So you need the expertise of the computer science. You need the specific expertise in the machine learning. You need expertise for the life science, chemistry, pharmaceutical science. So all collaboration is required. So that is also another challenge. But the big challenge is that the data. If you are really interested to do something in this field, so you have to identify the good data over the bad data, because the data contains the good data, bad data. If you have the good data, then you are in the right track. But if you develop your model with the bad data, by chance you develop model with your wrong data, then your prediction may be wrong, and that may fail somewhere in the drug discovery pipeline. So this is overall about the how we can implement machine learning, artificial intelligence in the drug discovery field. As I mentioned, this is the time, this is the uh, era of the machine learning. So if I remember in 90s, the early 90s or mid 90s, that time when the computer came in India, so that time the people knew of the very understanding about a computer, like Microsoft Excel, Microsoft Word, or PowerPoint. They got the job in a big company, and now they are in a good position, because that time the computers came in the market. That was the revolution that started that time. So similarly, that now this is the time of the revolution of the machine learning in the different fields, and especially in the drug discovery. So this is the time, if you know the machine learning, it is not like that you should have the very good expertise, but if you know, you to understand, so this is the time to get the job. The job market is widely opened, and this is the time to adopt the new technology, that is machine learning, in drug discovery. Now I'd like to explain a little bit about our company, that is SilicoScientia, what exactly we are and what we do. SilicoScientia is a startup, and it is a Bangalore-based company. We are working in the field of bioinformatics, cheminformatics, and definitely with the help using the artificial intelligence based approach, and also we are working in the artificial intelligence healthcare. So we are working in the different versatile. Also, we have our own platform, our own R&D we have. So I'll show maybe the next session. I'll show our own product. That is how. Yes, I will explain that time much bigger, because the people are scared. Especially the non-technical people from like pharmaceutical background, or maybe the chemistry background, the life science background, they scared about the computers, or they scared about um, the programming language. That hurdle we are trying to remove using our platform. So I'll explain that platform in the second session. So we are already in MSME and Startup India. So we are providing, these are the services like computational drug discovery or bioinformatics solution we're providing the artificial intelligence healthcare. Also, we're providing the skill development in the form of training. Suppose you would like to develop your skills in a different drug discovery, or artificial intelligence drug discovery, or bioinformatics, or AI in healthcare. So we are providing the training from 1 month to 6 months, depending upon your requirement. And also we provide academic research support. Suppose you have your master's project, you would like to do something in this new field or new in artificial intelligence, that also will help. Also the PhD thesis also we are helping here. Suppose you would like to do something in this field is in your PhD thesis, that's also we take care. So these are the overall thing actually we providing in our company. And now, as I mentioned, that this is the time of the machine learning in the drug discovery. So if you know drug discovery, if you know the computational drug discovery and how you can implement the machine learning, so the market is open for you. Because as I showed very briefly but the data is very, very long. That is, the investors or the companies are there jumping. They are crazy about it to invest in this field. So, and another thing is that in a there is a difference between the academic culture as well as the corporate culture. So we know the when the people selected in the company for the job, they are trained first and then they are ready for work. So that is another hurdle. So to overcome this thing, that is the student or the professionals, they should train in a corporate environment to get absorbed immediately in the company, and also how you can learn the machine learning, the drug discovery with the help, with the collaboration with the DI Patil uh, in DI Patil University, the School of Pharmacy and Research, Pune. The SilicoScientia launched 1 year diploma course in artificial intelligence and advanced diploma in computational drug discovery. So this is a 1 year course. It is already started, and the admission is going on. So if anyone would like to join this course, so he or she will be trained from the basic understanding of the computational drug discovery as well as the machine learning, like from the programming to how can you develop the machine learning on your own data by yourself. So that actually already started. So these are the course curriculum, like we're given a programming language, then bioinformatics as well as artificial intelligence, machine learning, then advanced level of the computer drug designing. So all these things will cover, and definitely you will have access the high performance computing system, the drug discovery software you will get. Uh, and it's a 1 year 1 year course, the 2 semesters, and it is this year it is the online mode, and the class started last week, and it's still admission is going on. If anyone would like to join, they may contact us, and it is definitely full time, and any students or final year students from B Farm or M Farm or any professionals or faculty members or research scholar, they can join. And it is fees, including all for the 2 semesters is 1 lakh for Indian students. So this is all about our course. So if I summarize this thing, that is yes, how the machine learning is impacted the computational drug discovery, okay? Because the computational drug discovery impacted in a large extent to the traditional drug discovery, and the machine learning will come into market. It enhance or it give the opportunity to the computational drug discovery to go explore the data in a wider range. So that is actually thing. And now, as I mentioned, yes, uh, this is the era, this is the time to learn, to use, to apply the machine learning in your domain. So thank you so much for your patience. If you have any question, we're open to take this.
Okay. So anybody has any queries or anything to ask from this session? You can unmute yourself and ask directly. Otherwise, you can post it in the chat box. Anything regarding AI and drug discovery or regarding the course from the session? You can unmute yourself and ask. Also, you can post it, write it in the chat if you have any queries. Anyone?
If you have any questions, you are free to ask.
Sir, I guess no one has any queries from this session.
Okay. But during the session, if you have any query, just you can ask, you can post your um, query in the chat box. We'll take it.
Okay. So, should we move ahead?
Yeah, so I will go for the next session. Yeah.
Okay. So, thank you so much, sir, for this insightful talk on transformative role of AI in drug discovery. I'm sure our audience has gained valuable experience and insights for how AI is transforming the computation-related drug discovery. So, let's begin with our second session. Now, Dr. Islam will take us through an exclusive session on SilicoXplore, next generation drug discovery suite integrated with AI based innovations. It is our next generation AI based platform for drug discovery. So, yeah, sir, you can continue further.
Okay. Thank you, my once again. And thank you for your patience, everyone, and your time. So now I would like to explain our platform and what is the objective of this platform and what are the things are there and what exactly we are doing and how you can incorporate in your research. So in my during my research career, or I talk different people from different background, like chemistry background with scientists, or maybe biology or pharmaceutical. So they are doing the drug discovery and they want to do. But the problem is that due to the lack of experience in the computational programming or the computational languages, or maybe the Linux platforms, people are scared to explore, or people are restricted themselves in a certain limited data. So in a normal machine, you can dock 10 molecules, 100 molecules, 1000 molecules, 5000 molecules. You can manage 10,000 molecules. Suppose you are very eager and you would like to do, you can dock 50,000 molecules. It will take long time in a normal system, but suppose you can manage. But suppose you have 50 lakhs molecules, 5 million molecules, then what will you do? Definitely, your normal system like your laptop or normal desktop, it can't help much. And also Windows can't help much. And it can help when you need the good programming language to handle those data. So that's why people, when we talk to different people, they are saying, yes, I would like to
Sir, you got muted.
muted now or from beginning?
No, no, sir, just now, just now.
Okay. So people want to dock, suppose, entire PubChem database. People want to dock entire Zinc database. People want to dock entire Coconut database. But they are very much scared about, because they can't handle that large number of molecules. That is the docking. Similarly, in the ligand base also, I would like to explore the huge number of molecules to get some proper conclusion. would like to explore the large chemical uh, space, but I don't have the expertise. That's the reason I can't go. So that actually felt us very at one of the area that is we can work.
Sir, again,
I don't know. Okay. Somebody is meeting by forceful to me. Just okay. Okay. So that's the reason we have developed one platform that is a SilicoXplore, and this is the end-to-end complete drug discovery platform. So here I will explain that is you no need any good server with you. You no need to understand the programming language. You no need to understand any Linux platform, but you can use as much as number of molecules you would like for your study. So in case of the docking, you can use millions of molecules. In case of the QSR modeling, you can use millions of molecules. In a docking, you can use the multiple proteins at a time. So all these things actually we solved, and we developed our the in SilicoXplore platform, and that is the beauty is that it is the cloud based. So everything will run in the cloud, and you will have to access through. You need your normal laptop, whatever you're working, and internet. So open the browser, open the SilicoXplore, and then you submit your data, and the rest thing will continue in the backend in cloud. So the people behind this thinking or this development, along with me, Dr. Sonali Thorat. She is one of the co-founders and directors, and she has expertise in chemical and pharmaceutical science. And also Dr. Shravanlal Bhomik is also the co-founder director of this company, and he is expertise in the biological and genomic science. So we are in a leading position. Along with that, these are the people behind behind this tool. They are contributing. They are contributed in the development of this tool in a directly or indirectly. So that I said that is the your drug discovery to manage the large number of data, screening issues, or the evaluation of the toxicity. So all these things, or maybe if you would like to have your good infrastructure with you, you need a good funding also. So those things actually are bottleneck in the drug discovery field. So those we have tried to remove in our SilicoXplore. So in the to solve the challenge, we need the DMTS capabilities, real-time support, research precision, admin filter, etc., etc. These are things that we need, and definitely whatever I discussed in the first session, that is the machine learning or artificial intelligence, that is the core part of the SilicoXplore. So you can develop your own model with your own data using this tool, and that model you can use to predict your new molecules or identify the new molecules, or you can differentiate whether toxic or non-toxic, or it is druggable or not. So this is a overall thing, that is a complete cycle that is you can do, and I will explain most of the tool here one by one. So what is the objective of this? It is reduce the gap between wet lab and in silico and the real-time discovery. And here the important thing is that we do some computational work, but when going to publish the data, we need the good quality, high quality data, or the illustration or the figures. So that is also challenging. So those people are publishing the data. I think it is our regular comment, that is, you have to improve your figures. The quality figure is not good. Increase the resolution. The figures are blurred. So those is also hurdle. So this, if you use this tool, so you will get the published to ready results and the illustration. And definitely, I'll and before ending the session, I would like to explain that is how it is affordable to everyone. Anybody is talking? Anybody is asking something?
No, sir.
Okay. And as I mentioned, you no need to understand any coding and any installation is required. Just you have to open your Google browser or the Chrome browser, type the website, and actually do it. So here, this is the interface actually, cloud-based platform SilicoXplore. So for target preparation, we have the SilicoPeptide. Notice target preparation. This is the peptide design tool based on the different properties or the complementary. You can design the huge number of peptide. So this is the machine learning based tool which can develop the potential peptide. And peptide nowadays is a very hot topic for as a therapeutic drug candidate. This is a protein modeling tool. Suppose you have the structure, you have the sequence, and you would like to put in the CD structure, you can use our protein modeling. Why we need this thing? Because sometimes the mutational analysis, that you have mutated some amino acids, and you can go for the modeling. So you can use the protein modeling tool, and very easy to use. So here, yeah, Maima, I would like to show one demonstration for maybe 1 or 2 molecules docking. So just prepare your data yourself after this. Okay.
Yes, sir.
This is a mutation inducer. So if you mutated your protein, how the impact on the stability will be there. So that can be predict here in the tool. So you just give your sequence, position where you would like to mutate, and then it will show due to the mutation what is the impact on the stability of the protein. Molecule design like Sketcher, this is very similar to IC's draw or ChemOffice like kind of thing. This is a de novo generator. So this is a machine learning based de novo generator. Suppose you would like to design the same new molecules for your study. So you come up with this, you can use this tool, and this is the machine learning based tool. It will generate a huge number of new molecules, and those molecules you can further screen and get the potential molecules. So we have already used this tool, and we have published a few papers recently. So mole conversion. This is a tool, like suppose you are handling 20 lakhs molecules, and 20 lakhs molecules you would like to go for docking, suppose in order of winner. So before that, you have to prepare the protein, now prepare the small molecule. So 20 lakhs small molecule you have to prepare. So it is not an easy task. And also in the molecules, there are a lot of conjugate is there, a lot of ions is there. So those need to be rectified. And also there is some valence issue. So this tool, it easily can convert your molecule or prepare your molecule from one format to other format by removing the duplicates, by removing the conjugate, by removing the ions, everything. And it is also fix the valence issue. All these things can you do in a very easy. Just upload your molecule, submit it. In few moments, you will get your prepared molecules. Fun fact: the pharmacophore is a very much important parameter to understand. That is how its molecule will be. So in that case, Yes, Maima, you should keep some data to show. Maybe two proteins or maybe four, five. Yes. Okay. So now here you can see if you have molecules, you can identify what are the pharmacophoric features are there, because pharmacophoric features are important for binding to the active site. So that will help you to understand what are the features, what are the functional group I need to delete or I need to incorporate. So that optimization actually helps with the form frag. SilicoCG to generate the conformers of your molecule, because you know the active conformer or inactive conformer. That is a very good option. Suppose sometime your docking results is giving very good affinity, but a conformer is an inactive conformer. So that actually also we can address through this tool, SilicoCG. That is, it will generate the huge number of conformers. Among these conformers, it will rank what are the possible active conformers for our study. Pocket finder, that is, suppose you have your protein, you don't know where the active site is. So it will identify all the available pockets in your protein, and it will rank, it will give the score, what are the score of this pocket, what is the score of this pocket, and that you can decide among these pockets which pocket I should take for my study. So it will give the score, it will rank it. So druggability score, you will get it. So that help you to find out the right active site. Silico scanning that I mentioned, that is, if you would like to develop your own machine learning model, so come up with your molecules, come up with your binding energy, biological activity, and then using those biological activity, you can develop, you give the input, the inactive and active data set, it will develop your own model. Sorry. It will develop your own model. So you can retrain until unless you get the validated model. Once you have the validated model, and that model you can use the predict of your new molecule or the entire database. So this is a very, very effective tool which can be used to screen the large chemical molecules, whether it is active or inactive or what kind of it is. QSR, you can develop your quantitative structure activity relationship model. It is a machine learning model. You come up with your data, and then also you can develop your model, retrain until unless you're satisfied, and then you can go for the prediction of the biological activity. And in this silico scanning here, what exactly I would like to say? Yeah. So here we can identify whether active or inactive. Here we can calculate the biological activity of the molecule. Similarity search. Suppose you have some good molecule, so standard molecule or drug molecule. You would like to identify similar molecules. So you can just upload your molecules and submit it. In a few seconds, you will get the similar molecule from the database from millions of data. Okay, so we have already our database. We have developed our natural compound database. Around 1.4 million compounds are there. That database also you can use to similarity search or exact search that we can actually do. So this is a molecular docking. There is a 4 docking tools right now we have. One is called ligand lock, then Vina, then diffuse dock, and dock. So here the beauty of this thing. So if you know the docking, so if you have the protein molecule, if you have the ligand, so what you can do, the one protein, one molecule, that is possible. One protein, if you know some scripting or you have some programming understanding, you can use the multiple docking, multiple ligand. One protein, multiple ligand. So that is there. But here, in this tool, the beauty is of this docking tool is that suppose you have 100 proteins, and lakhs molecules. So 100 proteins, 30 lakhs molecules. I would like to dock many to many. Each of the 100 molecules, 30 lakhs. So that means that that is 30 lakhs into 100. That much docking you can do here. So you just upload your protein, 100 proteins, upload your ligand, that is the 30 lakhs ligand, and submit it and shut down your computer. So the result will, the doc will be pop on the cloud, and at the end you will get the results. You can screen the results based on your criteria. So it is here 1-to-1, that is one protein, one ligand, one to many, one protein, multiple ligands, many to one, more than one proteins, one ligand, or many to many, that is many proteins, many ligands you can dock at a single click. So that is that here. This is the tool. Bina is the other tool. This is a machine learning based scoring functions is there. So if you know the docking has mainly the two things are very much important. One is the conformational ensembling, another is the binding energy calculation. So conformational analysis, the algorithm is there, like genetic approximation is there, random search is there. Similarly, in case of binding energy calculation, force field method is there, empirical is there, then knowledge based is there, hybrid is there, machine learning is there. So this is the machine learning based binding energy calculation is there. This is the another approach, and dock. This is also docking, but their algorithms are different. But the same thing you can do here, the 1-to-1, one to many, many to one, many to many docking at the same time. This is a diffuse dock, how actually your molecule automatically find the active site and binding at the active site. That also you can do. It is on kind of the validation of your docking. Then binding interaction analysis. So suppose you have one protein, you can upload your the online server, or you can use PyMol, or you can use Discovery Studio to check the binding interaction. Suppose you have 2000 complexes and you would like to check the binding interactions. You want to generate a table of the hydrophobic interaction, hydrogen interaction, water bridges, salt bridges, etc., etc. So what you can do? You have to go one by one. So here you just upload all 2000 complexes and submit it. It will do one by one, and it will give you the table at a time in a CSV format with their properties, and it will give you whether it is a bad or good, or it is we can consider it's not. So all the molecules, see, you can see here the molecule. Also you can download this molecule from here. This is a toxicity prediction tool. So you would like to know that is whether your molecule is toxic or non-toxic in nature. So this is the AI based tool. You just upload your molecule in a small format. Suppose you would like to upload 20,000 molecules. So upload it. It will show you. It will generate, and it will show you what are the molecules are active and toxic, or what are the non-toxic and moderate toxic. And this will give you the pie chart, and also you can download the data, entire data, or all toxic molecules or non-toxic molecules. All these things you can download from here. This is a physicochemical property. So you just upload your molecule, and I think more than 50 physicochemical properties you can calculate for each of the molecule. You can download it, and this will help you to decide what are the molecules are good in nature, or what are the molecules should not be considered for our study. Molecular dynamics is a very, very effective tool in case in the drug discovery field. And problem is that you need to understand some Linux platform to run this MD simulation. Yes, Desmond is there, but there's some limitation is there. But whatever the Amber or the Gromax is widely freely available tool, and it is most trusted tool for the molecular dynamics simulation. But we are scared about to the command line to edit the files and all these things. That is a big hurdle to do the molecular dynamics simulation. So we have just solved it. You have to just upload your ligand protein and select what is the force field you would like to do, and just how long you would like to run the simulation and submit it. The, everything will be performed in a backend. You don't need to do anything. And once it is completed, you can go for analysis. You can calculate the RMST, RMSF, SASA, free energy landscape, PCA, DCCSM, lot more. And you can plot the data, and you can download the plotted data in a high quality, high resolution data. So you can plot the multiple graphs at a time, the same graph and multiple molecules, or single also. So this is actually nothing. If you would like to do the molecular dynamics simulation, if you're scared about the programming, nothing, or you're scared about the Linux, or you don't have this, because another thing is that the molecular dynamics simulation requires the GPU. Because CPU, it is possible, but it will take huge time. It is time consuming, it is resource consuming. So that you no need to worry. You just submit your simulation. Everything will run our cloud computing GPU system, and you will get ready data. Similarly, we have the DFT calculation. You will just upload your molecule, or you will draw your molecule, and you will get the DFT along with the other parameters. DFT figure, homo energy, LUMO energy, then dipole moment. All these things you can get, and you can save for your report or for your manuscript. So this is also there. You have nothing to do, just draw your molecule and submit it. Like first QC, some QC file is there, that data you would like to analyze. So you can upload your first QC file, and all the plots, all the analysis will be done here, and you can see, you can interpret your data. Visualization, that is in a different orientation of visualization you can do for your molecule. Rescoring, this is a very, very important tool. It is actually very much helping to decide about the good molecule. So you have docked the molecule. You know this is docking is very good, and you're getting good binding affinity, but you don't know whether it is a false positive or not. So this rescoring tool will regenerate the confirmation of your poses and calculate or predict the binding affinity. Okay, so if you are getting the better binding affinity, that means you are okay. If you are getting the lower binding affinity compared to the original one, that means it is the false positive. So this will help you to improve or help to select the better pose, better orientation. Also, it will help, that is whether you are taking the false positive or false negative data. So this is very, very important tool for selecting the molecule from the docking. You can use here nucleic acid small molecule or protein small molecule complexes. That is, I said, a post docking. Once you have the dock of your molecule, like multiple protein, multiple ligands, now you'd like to screen out the molecule, so you can just select it and use the binding affinity, and you can get the molecules with their complexes. Apart from that, it is coming the SilicoDock, another docking tool is coming very soon. Silico structure relationship tool is coming. Then mutational age calculator is coming, and definitely the Bioinformatics analysis tool is under process. So these tools will come very soon in this platform. Apart from that, we have also our bioinformatics platform, that is named the Bioforge. So in case of the computational drug discovery, we are dealing with the small molecule, protein, all this thing. In the bioinformatics, we are mostly working with the sequences or other kind of data. And that data is a huge data. So to handle those data, because for one analysis, it may generate around 100 GB or 200 GB or more. So those data handle is not easy. That actually we overcome it, and we have our own platform like Bioforge. Here, RNA-seq analysis already done. So this is already launched, this tool. You have to upload the data and select one by one. It is actually calculate and it will interpret and give the data, the quality. Also, we have the WES, the whole exome sequence analysis tool is here. These two pipelines are already incorporated, and more pipelines are the process that will come very soon. So now the big question you may ask, what is the licensing policies? So we are so if you are to go for the research licensing, that will be different. If the academic license, that will be different. Corporate license is different. Individual, if you would like to do individually, suppose I am the researcher, or I am the scientist, or I am the student, I would like to go for the individual license, that also available. Group license also available. Suppose a research group can get a license. 2 or 3 more research group can take license, or a group of students can get the license. And also more flexibility, because sometimes it is because if you go for the other tools, they are very restricted. You have to go for at least 1 year licensing or at least 6 months licensing. But here we are very much flexible. Go for the 1 week to few weeks, that also available. 1 month to few months. Or long term, maybe 1 year or maybe more, that also we are providing. So just I would like to, for your information, I'd like to inform you that the SilicoXplore already taken by the Nagpur University, the pharmaceutical department. They took license for 5 years with us. So they already delivered last March. And they are working, they are using that platform, the entire SilicoXplore platform, and all the calculations I mentioned, they are submitting job, but all the things are calculating in our cloud. So it is a hassle-free platform. No need to bother about anything. So it is actually user-friendly, what I can say, kind of thing, tool. So if some, maybe we'll show one demonstration, because around 26, 27 tools are there in SilicoXplore. So it is not possible to show you all the tools, but maybe 1 or 2 tools Mahima will explain. So what is actually here? That is it integrated AI and physics-based method. Physics-based method is that not out a question, but when it integrated AI with the physics-based, that actually get a new dimension, and it is cloud-based and is scalable and user-friendly. It accelerates the drug discovery process, and ideal for both academia and industry, and also it supports the education innovation. So thank you so much for your patience, and I am happy to accept a question related to AI in drug discovery, related to the platform, related to licensing, all these things. I'm happy to accept your question. Thank you so much. Yeah. If you have any question, just unmute yourself and ask, or you can type in chat box. Anyone has any question?
Any questions, anyone?
No question from anyone? Yeah, we'll talk maybe after this session. Okay. May I just show on talking using this tool. And then we'll move to the last session like discussion forum.
Okay. So is my screen visible?
Yes.
So this is our platform, SilicoXplore. And here you can see there are different tools, docking tools, analytics tools, and prediction-based tools. So these are the docking tools, 4 docking tools we have with different algorithms. Then visualization tools, protein modeling, toxicity prediction tools, conformer generations, and these molecular dynamics also we have. 26 tools currently we are having in our drug discovery suite. So for now, I would like to show you a short demonstration of our tool Ligand Lock, which is a molecular docking tool. And here we have automated every process. If somebody has been used Autodock tools or Autodock Vina or any different tool for docking, they might know what are the steps we have to do. And there is a lot of manual work which we need to do. So in this tool, we have automated all processes from protein preparation to the post-docking analysis, all the options you get here itself. So let's begin. Let's begin with the first step, that is the protein preparation step. So there are 2 options for the protein preparation. One is you can directly upload the file from your system. For instance, I am taking this PDB file to ICF. Okay. First, we have to create the job ID. Without job ID, it won't work. Okay, just click up, write the name of your this thing, your demonstration, and just click on the plus sign. So your job ID will be created. Then first option to upload the protein from your system. If you have any files with your protein, you can upload it. So here you can see the protein which I have uploaded from my system. Also, you can see the amino acid structure of what this protein contains here. Now, one protein we have uploaded from our this thing from our system. Another protein, if you want to upload, we can, another option for uploading the protein is directly fetching from the PDB database, protein data bank. Suppose I am entering the PDB ID and just click on the load button. So this is your protein. Second. If you click here, you can visualize the protein. So now we will go to the preparation step. So first we will prepare this 2ICF protein. If you click on this properties symbol, you will get all the properties which your protein has. How many chains the protein has, what are the ligand atoms present, what are the ions, any cofactors or any heteromolecules are present into your protein. All the options, all the details you will get here. So from here, we will keep whatever we want and we will select the chains or ligands, ions which we don't want in our protein. So we will keep the chain A as this protein only contains a single chain. Then there are 2 atoms, one is NO3 and one is YCF ligand. So we'll just keep only one ligand and check the second one. Also, we don't need any ions. Also, we will delete the water. So whatever is checked, that will be deleted. So save selections. Okay, so now for the first protein, we have selected the properties. Now we will uncheck the first one and check the second one to prepare the second protein. Okay, so now if we will click on the properties, here you can see this 5G15 protein have two chains, a single ligand, and multiple ions are present. Magnesium ion, SO4 ions, and water are present. So we will, for instance, we just need chain 1 for docking. So another chain we will delete. All the ions also we will delete and just save this selection. Okay. So now the next step is to prepare the protein. So we will select both the proteins and we will click on the prepare selected proteins. So it will ask that whether you have to prepare both the proteins or not. So we'll just click on yes, prepare them. Okay. So here you can see the preparing. It can be seen over here. Just a minute. Okay. Yeah. Sorry. So after your protein has been prepared, pop-up will come with the message that preparation completed. So 2 proteins have been prepared successfully. So both the proteins which we have uploaded that are prepared. Now protein preparation step is completed here. Next, we move on to the link. So is somebody asking something?
Molecular simulation, actually, it is time-consuming matter. So what we can show you, that is, yes, how can we submit, what are the parameters you need, that's all we can show you. Yeah, definitely. We'll show you, that is what, how can you submit it. Yeah, definitely.
Okay. Yeah, so next step is the ligand preparation. So just upload your ligand files from the upload ligand file option. Now these are my ligands which are in SDF format. So once I upload, I can visualize or I can see the uploaded ligands here. So here you can see in the uploaded file, you can see how many ligands are present. There are 5 ligands in this file. So one by one, you can just check their representation and visualize them. So what we are going to do, we are going to prepare all the 5 ligands together. Okay. So these are some options which we can check. If you have a file with 2D conformation, 2D coordinates of your molecule, so that you can convert directly here by checking the option of generate 3D coordinates. And if now our file is only already in 3D, so we'll not check this option. We will add hydrogens. The pH we can change, and also we will add the charges. So here all the options we have selected for preparing the ligands. Now just we will click on prepare the ligand option. So here it can be seen that within like less than a second all the ligands have been prepared. So there is no limitation, like you can upload a large number of compounds and prepare them, like 1,000, 2,000, 50,000, 1 lakh molecules also you can prepare using this particular option. So protein preparation done, ligand preparation done. Now the next step, that is the grid generation. Okay, the auto grid generation means what we are doing, we are telling, like we are giving the parameters to for our ligand to go and talk. So that how we are going to do. So there are different options here. First, we will click on the show protein. So these are the 2 proteins. which are selected or we have prepared just now. Then what ligands are prepared? So this ligand also we have prepared. You just have to check that which protein you have to take for docking. If you wish to go with only one protein docking, so you can just uncheck the another one and you can keep the one. Now we are going to do two protein, and with both the protein we are going to perform the docking one by one. So first 5G15 protein, we will define the docking box. So these are the options by which we can select the grid parameters. Active site detection. First is co-crystal ligand. If your protein has a co-crystal ligand attached in its crystallized structure, so we can auto detect it from here itself, and we can build our grid uh, on it position. Second, if you have the co-crystal ligand downloaded into your system, so you can upload the co-crystal ligand and tell this tool to build your grid on it. Then cavity detection, using the different cavities present in your protein. If you have some particular important cavity which you want to target, so that option we can use and build our grid on that particular cavity. Also, you can pick the amino acids from your protein sequence, amino acid sequence. If you have some key residues into your protein structure which you want to target, and you want to dock your ligand into that particular site, that also we can use. And also you can manually give the coordinates. So for now, we are going to select the first option, that is the auto detect of the co-crystal ligand. So first we have to visualize the protein. So here you can see there is a co-crystal ligand present in our protein. So we'll just click on the auto detect and define docking box. So you can see the coordinates, that is the x coordinate, the z coordinate, or the coordinates have been auto detected, auto calculated from this option, and just we will preview. So if you can see, this is our docking box around our co-crystal ligand, this one, the dotted one. Now you can see it clearly. So this is the portion, this is the grid we have generated using the auto detect co-crystal ligand option. So we can just save it. And from here we can save the config. Okay. Now for the second protein we need to prepare. Now this is our second protein. Again, we will select the first option, and this is the coordinates for our second protein and the dimension of the box. By clicking on the preview, you can see the second grid and just save the config file. So you will get a notification the config file is saved. Now for both the protein, our grid parameters are ready. The active site detection is done, and the ligand is also prepared and we have selected. Now what we will do, we'll just go and click on the run docking option. So docking job has been submitted. If you can see clearly at the very bottom, your job has been submitted. It will take few minutes for us to complete the docking. So we have selected two proteins and one set of ligands, means this ligand set will be docked with both the proteins. Few minutes it will take to dock.
If you have any question in between, you can ask.
So next will be checking the results after the docking is completed. So in the result tab, you can just check like whichever ligands docking is completed, it will come here. So first we have to select the protein, okay, which we want to check the results for, and also the ligand set which we want to check for. And then click on the fetch result option. Now you can see 4 ligands are completed till now. Also, the ligand name will be displayed.
Click on the ligand. Click on ligand, first ligand. So you can see here you can rotate and dock like that. You can see.
So this is the dock ligand and this is the binding energy, binding affinity of that particular ligand. So total 9 poses have been generated. So one by one you can check all the 9 poses of your ligand molecule. Also, once we check the ligand one by one, the binding affinity also is displayed over here of that particular ligand. So the highest is minus 7.6. So I guess the docking is completed for the first. Yeah, all the 5 ligands have been docked successfully using this protein and the ligand. So one by one we will just check. So here you can see all the ligands, all the 5 ligands are docked into the binding cavity which we have selected, the active site, the grid which we generated, and that side all the ligands have been docked successfully. So you can download this results also. Also you can see the binding energy graph of each pose of that particular ligand with the protein. So first pose, binding affinity, second pose, third, fourth, and likewise. So you can also download this graph also. So let's check with the second protein, that is 2ICF protein. So 2 ligands completed for this protein. So the highest binding energy is minus 9.8.
Go to right side here. From here you can plot. You can show the plot. So the binding energy of the different poses. Or also you can download a complex pose that you can do.
Okay, so all the docking is completed. So the pop-up shows you.
So here the binding and interaction of this protein you can see, and hydrophobic interaction, hydrogen interaction, number of amino acids you can do. You can download the CSV format also. Excel file. So this way you can do docking. So since it is only 2 and 2 molecules or 2, 3 molecules, it is it won't what is going on. But if you have the huge number of molecules, so then you can understand exactly how it is happening. Easy to do this. Okay. So then this is all about the docking. That is very easy when you submit multiple protein, multiple ligands, thousands of ligands you can submit and just relax. Okay. Now go to the main page. So this way actually you can generate a lot of things. Just deactive ones. Just create job ID. Okay. Now just draw toluene or maybe phenol, benzene and one bond and add extra group. Draw it. One bond up and one single bond. And just put hydroxy group. Oh, right hand side O. Omkar, you need to change the any states or auto is fine. No. Control Z, Control Z, Control Z. Now go to the CS3, top of CS3. Press, no, it's on share, yeah. Click. Omkar is there, no?
Parts is there, I guess.
Up.
Yes, sir.
Do you need to change the functional or basis sets?
No, just keep the auto select.
Okay, just run the calculation.
So all the calculation.
Yes.
Yes.
So depending upon the molecule size, it will take time. Very simple molecule, so I think it won't take much time. Go down. So here you can see the running. The optimization of the geometry is running. So you will get after that all the data very shortly. And after that, we will show you the interface of molecular dynamics simulation because we can't submit right now. But we can show you how what are the parameters you need for the simulation. For institution, you want for the full demonstration, so we can do it maybe online, and if you are interested for purchasing and all these things, so we can come to your institution also and we can demonstrate it. Or if you want one line to each of the tools that are how you can apply it, what are the application, the details theoretical thing also we can demonstrate if anyone saw. So thing is that once you submit the data, you can go, you can shut down your machine. You will get the notification in your email that is job has been completed. And also, if you come back and log in, and then you can fetch your data in the job ID. So it is not like that you have to keep your computer on. You just submit or shut down your machine, and then you come back maybe one hour or 1 week or 1 month. That does not matter. because this is computationally expensive tool. It requires a lot of calculation, quantum mechanical calculation. So that's why it's taking time. Ah, Last is coming running. Yes, special push. Anyone in this crowd are working on TFT? Anyone? Any tool maybe? You have used any tool for calculation of TFT? Yeah, go down. Completed. So this is the homo energy. You can just rotate it. And you can just or opacity or all these things you can change. This ISO. Yeah. And then ISO value just 0.8. So go up a little bit. This is the homo, actually homo, sorry, lumo. Just click on lumo. Here you can see the electronic surface area, ESP. And then you can change your according to your orientation. You can see like this. Go down. Go down. So here homologo energies you can check. Reactivity descriptors you can check. Then molecular charges you can check here. Go down. So all this data for this phenol you can get. And yes, go up. So all data you can download or the figure you can download together. Okay. So this is about another year. Okay. Now just go to the okay, here the two options are there: protein ligand simulation and protein simulation. If you go for only protein simulation, then you can say protein ligand simulation. Energy, if you click on just create job ID first. So job ID created. Energy minimize. Click on energy minimization. So these are the default parameters. So advanced. So advanced here. So these are the default parameters. If you would like to change it, otherwise save it. Go down. So basic, and save it. save changes. Similarly, ions, you can, if you want to check a change, that is fine. Someone actually asked you, yeah, Karishma. Okay, just for you, just for your request, I'm showing you. So how it is easy. So you can just save changes. Then equilibration, how long to go for equilibration? So like this, you can change the end step, or here by default 3 nanoseconds is selected. If you go to advanced thing, then you will see more parameters if you'd like to change. And go to the basic and save it. And similarly NPT, and yeah, save it. And then MD, that is production phase. So here you can change the production phase. I think is by default it is 10 nanoseconds selected, and you can save it. So all the parameters are saved. Okay. And now here select the force field. So the first one is charm 36, so you can suppose 6 here. And here you can use the Swiss param or anti-chamber to create your topology of your ligand. Okay, that you can select. We are not going to run right now. You can use Swiss param or you can use the anti-chamber for the ligand topology. And if you have the ligand topology already with you, you can just upload here. First, upload the protein here, and then each of the ligand files you can upload. Okay, all this file here, just upload it. Go down and just submit the. Once you uploaded all this file here, just submit the simulation. So your job finished. You have to nothing to do. Like if you anyone is expert know about the simulation in Promax, so first you have to create the PDB to GMX for the protein topology. Then you have to edit the protein file, and you have to edit the topology file. You have to make the complex. Then you have to generate the box. You have to solvate it. Then you have to neutralize it. Then you have to go for minimization. Then you have to go for equilibration. Then the before equilibration, you have to go for the restraining, the equilibration 2 types, and then submission the final production. But here you nothing to do, just set the files and just upload the files here and submit it. One by one, automatically everything, editing, submitting, all will be done there. And once it's completed, you will get the message. And if you go to the analysis, so here you just give the TPR file, and here the XTC file, select it, or you can give the path also here. Here in the bottom you can see the net server path. Suppose it is file is big, so you just give the path here and run the analysis. If you run the analysis, automatically it will center, it will get RMST, RMSF, SASA, FEL, PCA, all these thing calculation will be done, and the plotting will be done, and you will get the plot after analysis. And another option is there here. Once suppose you have 3 molecules for simulation, you complete your 3 molecule simulation, and then you plot it. So in one plot, the 3 molecules plotting will be done. For that is required for the publication, all this thing. So that also can be done. So this is actually the very user-friendly simulation. You no need to write any command, no need to edit any file, anything. You just upload the files of your protein or your ligand and submit it. And analysis is also very easy. What are the analysis you would like to do? Okay. So this is about the simulation. Go back. Anyone you would like to see any other tool like protein modeling, toxicity, QSR, these things will take time actually, so I'm not going to all these things. Mutation inducer, pocket finder. Pocket finder we can show very quickly. Go to the pocket finder. Create a Java ID. Upload. So here you can identify your active site. Upload the protein. Okay. So here you can just upload another protein. You can upload multiple. Kashma will discuss after this. Actually, we'll discuss the discussion forum. All these things we'll discuss. Okay. So 2 proteins we have uploaded. Now we'll go for the pocket finder. So select the first one. Find pockets. So you can see, go down. You can see these are total. These are the pockets. Left hand side total 24 pockets found. So select one pocket there. Like 10 pocket. So you can see, go up, see the right hand side, the position of your pocket. You can see. Just zoom in. You can rotate. So this is there, and go down again. Select any two, 3 proteins again. 2, 3, sorry, pockets. So here you can see in the right hand side bottom the pocket number and the pocket scores, like pocket score, drug score, alpha sphere. All these things you can do. Yeah, Sadhana are coming actually. We shall recording or expression of an active drug and polymer. I'll okay. So here go down. The position you can see and go down. So this value will help you to decide which pocket I should take. Okay. Now go to the second protein, unselect the first protein and go to select the second protein and find the pocket. So you can see here 44 pockets are found in the second protein. You can click on there. And you can show the pockets, all these things. So here you can see 2 different right hand side. You can see the 2 different proteins, 2 different proteins, and that their pocket numbers and their score. So here comparative analysis you can do, and it can help. Select a few more pockets. The top also. From first protein. Few more for some pockets. So that actually will help you to see. And here the plot is there. Just go to the plot. Go down. Right hand side. Go down. Compare pockets. Compare pockets. So here in the graphical user interface, you can see among these pockets, what is the pocket score, what is the drug score, what is the alpha sphere, what is the volume, MC volume. Go down, give it in the graph. So here in the x-axis, alpha is in the x-axis, you can see the pocket name, or if you go to keep your mouse, you can see there. Go down. number of atoms, hydrophobic scores, all these things are there. Okay. So these comparative analysis will help you among these two proteins, what are the active sites I should take for my research. So that is very much helpful to identify the right pocket. You can download this plot also, comparative plot. And also these values you can download using the CSV format. Okay. So we shown here two proteins. You can upload 10 proteins, 20 proteins, 100 proteins together, and you can find a binding pocket at a time. You go up. Go up. So here you can see the visualization also. So you just hide the second protein. The eye icon. So here you can see, if you rotate this protein only, you rotate and zoom in. So you can see the position of the each pocket and their size you can see from the value. Okay. So now we'll discuss your issue and charisma also issue in the next discussion forum. Okay, so this is the tool, one of the tool for the finding. So similarly, the conformance generation, pharmacophoric generation, then protein modeling, that is homology modeling is there. Then rescoring is there. Silicopeptide, de novo generator, that is a new molecule you can generate. Similarity search. Okay, go top. Okay, database. So this is our database. It contains 1.4 million compounds, and you can search by name, Smiley's ID or formula, or if you go to the advanced option. So here you can search by the Smiley's, or you can draw a structure, you can upload a structure, and the options are substructure searching. That is, if you want to keep some, suppose you have one fragment and you would like to see the fragment in all the molecules, substructure. Similarity searching, that is the percentage wise, the tiny bit of coefficient. Exact match, suppose you have given one. We'll talk about it. Then you go for the exact match. So suppose you have given the structure, the exact similar structure you can get. So here actually get, and also you got the molecular properties. So suppose you would like to find some molecule with the certain values of the molecule, like hydrophobicity like this, molecular weight like this value, drag likeness, etc. This polar surface area, BBB. So all properties you can select. Toxicity parameter also you can select. So if you select these properties and submit it, it will retrieve the molecules from the database. Okay. So this is about our database. So this is just I show you the glimpse of our so this is our actually the glimpse of our tool. So much more things are there, and a lot of machine learning applications are there. Lot of machine learning models are running behind it. So that is there, and similarly we have the bioinformatics forge that is under development. Few things are there. So if you are anyone interested, we can show that also. But since it is the data analysis is much time consuming. So if anyone interested, we can show that also. So this is all about actually our tool. So now we'll go for the last session. Just if you stop sharing my and this is open discussion. So this is not like the question answer, your thought. Uh, this is more or less a discussion to each other. Your opinion, what do you think, what might be like this thing, so that we will discuss, and whatever the questions you have, that we will try to answer. Okay. And one important thing, now we're going to give you the feedback form in the chat box. So everyone, please fill the feedback form, and once you have, whoever will fill the form, they will get the certificate via email. Okay, it will take maybe 1 week or 10 days to get certificate. So kindly fill your feedback form. Okay, now. Charisma, you asked about it is freely available tool or paid. It is a paid one. Definitely, it is paid one because two things are there. One is that we have developed this tool from our organization, and the second thing is that all these things are running in cloud. So we are going to provide cloud. So that's why it is chargeable. And if you're asking what is the charge, so that depends upon your requirement, for how long you want, for how many licenses you want, and what are the things you would like to run. Definitely, if you want to molecular dynamic simulation, the cost will be much more because it is running the GPU, and GPU cost is high. But if other, it is very less if you're not considering the molecular dynamic simulation. So the licensing policy, depending upon the organization, your research objective, and for how long you want, for how many licenses you want. So that we can discuss. If anyone interested, you just email us. We may discuss one-to-one. Sadhna, that is which tool can be used for molecular modeling of this version of active drug and polymers? I'm not actually getting the question properly, but you can unmute or you can talk, Sadhna. Uh, uh, but what actually polymer drug conjugation you are talking about, if I'm not wrong? Am I right? What exactly you're asking, Sadhna? You can unmute yourself and you can ask. So we are actually working on the polymer thing because we are for material science we have not launched yet, but we are working and the work is going on. Definitely will do it. But the particular question, that is, if it is a drug polymer conjugation, this tool is not incorporated yet, but we do practice this thing. Yeah, Vikram.
Yes, sir. Sir, as per polymer design and synthesis process, so this in our platform, DFT is also useful, like for 3D printing of polymers. So polymer contains so many bonds, like it is a dimerization or conjugation bonds. So we can define its elasticity and softness of polymer for the 3D printing and various type of analysis. So I think our DFT tool also helpful in the polymer design and research purpose. So like DFT, also we can dock the polymers through our docking tools also for the any particular protein and its polymer process and also. So I think in the 3D printing, it is very useful for designs.
Okay. Okay. Thank you, Vikram, for the explanation. Sadhna, I think hope you got the answer. If you have any further questions, you may ask. Okay. And yeah, I was we are not connected to Med DRA. So whatever the database, it is no, it is not connected to the Med DRA. Okay. Anyone has any query?
Any questions, anyone? The feedback form is already shared. The link is shared. Kindly please share your feedback and improvements.
Yes, we have our own servers. That means yes, because we controlled by us, so it is highly secured because our first priority to the security of the data of the client. It's so definitely it is. And there's a multi-layer firewall system is there, so data will be secure. Yeah, nice. Can you tell me about your tie-up with RTMNA? Yeah, RTMNA, that is our softwares they have like they took the license for 5 years from us for the SilicoXplore. In this field, I have used Schrodinger, BioVia, Reset, MOE, ICM, long time. Problem is that they will install it, they will give initial training, maybe 2-3 months, and then after that they won't provide any support. And the problem is that also for Schrodinger, they won't give you the updates. Whatever they are, they won't provide another licensing, might be 1 year or 3 years package. Similarly, the commercial the research license is too much expensive for all of them. Like Schrodinger, BioVia, Reset, everything. Their academic license is less, but academic license you can't publish the paper for only for student. So we have the tie-up with the RTMNU, and we have provided a research license. We have agreement that they can use and publish the paper. They took 20 tokens from us, and we have demonstrated last June, and we are providing the update. Whatever the updates are coming, every 3 months we are updating the updates, new tools, new algorithms are incorporating. So we are providing, so this will continuous. And also for the training, whoever will take our software will provide that is one line, that is every 3 months online training, and once or twice in a year the on-site training. That actually, upon that, we are available 24/7 for the technical. Like we have the ticket raising system is there, and also we are available over phone. So that is there actually. This is actually the collaboration with the RTMNU. I hope you have got the answer. Anyone has any question or your suggestion? You can talk. This is the discussion from. This is not like question and suggestion. We would like to know about your view about this field, that is how drug discovery impacting through the artificial intelligence, and what will be the next, how it is going to change. So please unmute yourself and you can discuss. Everyone is scared, that is what I should say. This is openly, this is nothing actually, this is just open discussion. Definitely will, Manish will go for more webinars. And last month, I think we have conducted workshops, 2 or 3 workshops we have conducted. And we have our internship program, like it is I mentioned, a skill development program. So anyone who would like to learn, please contact us. There's some charges of their charges will be there. So that also we provide. We trained 1-to-1, like, or maybe batch also. Manual docking or traditional way of docking will be replaced. What do you mean by the manual docking or traditional docking? That actually, docking. Ayush, could you please explain what is the manual docking and a traditional way of docking? Traditional, that means you're talking about using the tools. Okay. Yeah, I got it. I said that you are perfectly right. That is the tools right now. That is actually the physics-based method mostly. Like all of the algorithms are there, like Glide they have, Vina they have, then Bioinformatics they have. So these are physics-based method. This is the established method. So if the AI-based method won't, I don't think so. It can replace. But when you merge the physics-based methods and AI, so that will change the dimension. Because only the AI-based, there is a lot of till now. Maybe I don't. Maybe in the near future it will improve. But till now, the problem is the trust issue, trustability. How your model will perform, whether it is a node performing, whether it is the false positive predicting. So that is there. So, but if you combine the physics-based and the AI-based method, then definitely it is very much helpful. And why it is helpful? Because you can incorporate huge number of molecules or huge number of data with a shorter time, with a less computational resources, and that is required. So that is one of the objective of our SilicoXplore. We have the physics-based tools, physics-based algorithms, and also we have the machine learning-based algorithms. And both we have to combining, and we can explore the huge number of data. So replacement, it is maybe possible. Some people may think that, but according to me, it is not. Better to combine both and do much bigger work. Okay. Please, anyone, your view, your query, your suggestion. We'll wrap up very soon. And all of you, please fill your feedback form so that we can provide you our your certificate. And also, Maima shared our social media link, like workshop, webinar, or any training program. All this will get update from our social media. So you just you may follow it for new updates. No suggestion from anyone? Okay. So thank you so much for your interest. Okay, ma'am, I just connected.
Yes, sir. Okay. Yeah. So as we come to the end of today's webinar, I would like to extend our heartfelt thanks to Atul sir for his valuable time and sharing his knowledge on various fields of drug discovery as well as AI-based drug discovery. So a big thank you to all the participants for joining and sparing their valuable time with us today and making this session successful. So just a quick reminder to fill the feedback form compulsory, and those who will fill the feedback form, we will be sending our your certificates soon. Within 7 to 10 working days, we will send the participation certificates. Yeah, forward to hosting such more future events. Until then, stay safe and keep learning. Thank you. Have a great day ahead, everyone.
Thank you.